Subscribe my channel and press bell icon to get all notification.
Hello students, in the previous video we started our topic, that is the cell cycle. We discussed about what changes occur in the G1 phase, S phase and G2 phase. यहाँ तक हम कर चुके हैं. Now what happens in the M phase we will discuss in detail later on, but before going on to the M phase, let's discuss some more thing about the interphase. Here we discussed that when a cell enters into the interface, जब एक cell interface में enter करती है, after that it will divide and it forms the two daughter cells. That concept is clear to you, that one parent cell and enter into the interface, it's grow, it's do all the preparation, and then after M phase it forms two daughter cells. Now let's see what happens. Suppose this is the two daughter cells which are formed from which are formed after at the end of the M phase. M phase के end के बाद ये दो daughter cell बन गई है. We are saying that this is a cyclic event. Cell cycle is a cyclic event, and these daughter cell will again enter into the interface and they will divide further. Okay. Now, but what happens if these daughter cells will do not divide further? अगर ये daughter cells आगे जाके division ना करें, तब क्या होगा, तब वो कंडीशन क्या होगी? सो वी विल डिस्कस अबाउट दिस कंडीशन इन दिस वीडियो. When these daughter cells do not divide further, this is their last cell division, इनका last division होगा, तब क्या होता है उसके बारे में हम देखते हैं. This daughter cell, when they do not want to divide further, they will enter into the interface. ये interface के अंदर enter करती हैं. When they enter into the interface, they grow. They increase their size because their size is not as similar as the parent cell, so they need to be grow. यहाँ पे इनको growth की जरूरत पड़ेगी. They enter into the G1 phase, or we can say that they enter into the interface, and into the G1 phase they grow, they increase their size, and after that they come to the they enter into the another phase. They do not continue the G1. They enter into the another phase, and this another phase we will call these another phase is G0 फेज. तो यहाँ पे हम इसको ऐसे दिखा सकते हैं. सपोज ये एक सेल थी, सेल ने इंटरफेस में एंटर किया, एंड इन द इंटरफेस इट इंक्रीजेस इनटू द साइज. साइज अपना इंक्रीज करेगी पेरेंटल सेल्स के जितना अपना साइज एक्वायर करने के बाद इट एंटर इनटू द अनदर फेज, एंड दिस फेज विल बी G0 फेज, और वी कैन से इट G0 फेज. So it will enter into the G0 phase if it don't want to divide further. So let's see. When it enter into the Z0 phase, this Z0 phase is known as inactive phase. This Z0 or Z0 phase will known as inactive. This is not inactive, means actually it's not inactive कि ऐसा नहीं है कि इसमें सेल के अंदर कुछ भी नहीं होगा. It is inactive phase only for division. सिर्फ फॉर डिवीजन. ये सिर्फ डिवीजन के लिए इनएक्टिव है. यहाँ पे सेल अब सिर्फ डिवाइड नहीं करेगी, अदर सेल एक्टिविटीज विल रिमेन एज इट इज. सबकी सब एक्टिविटीज चलती रहेंगी. So in this way we can explain the Z0, but in this way if you represent the Z0 phase in this way, that it creates a little bit confusion. What happens, इससे क्या पता चल रहा है? इससे पता चल रहा है कि M phase के बाद सेल ने एंटर किया G1 phase में, size increase किया, and then it enters into the Z0 phase, and after Z0 it will enter into the Z1 phase again. वो फिर से Z1 में आ गई है. तो ये चीज यहाँ पे थोड़ी सी कंफ्यूजन क्रिएट करती है. टू अवॉइड दिस कंफ्यूजन व्हाट वी कैन डू, इस कंफ्यूजन को दूर करने के लिए हम क्या कर सकते हैं? वी कैन शो इट इन अनदर वे. हम इसको एक दूसरे तरीके से भी शो कर सकते हैं. व्हाट विल बी द अनदर वे? दूसरा तरीका क्या होगा? दैट वी मेक अ साइन लाइक दिस, दैट हम यहाँ पे इस तरीके से दिखाएं that cell is exiting out from the interface. In this way we can show that the cell is now no more dividing. It's not dividing further. It's enter from the cell cycle, exit from the cell cycle. Cell cycle से exit कर चुकी है. And this cell which exit from the cell cycle and it enters into the phase which is Z0 phase. And in the Z0 phase, the cell which enter into the Z0 phase, the cell is known as differentiated cells. Differentiated cell. So we can call the cell which enter into the Z0 phase, we can call this cell as differentiated cell or the specialized cell. Means अब cell specialized बन गई है for do its function, for its working. It's no need to divide it further. So in the Z0 phase, when the cell enter into the Z0 phase, we call the cell as a differentiated cell or the specialized cell. So this is the first condition which we have to understand here. Now, let's see what happens. In some cases, the differentiated or specialized cell also need to divide further. कभी कभी ऐसा होता है कि जो differentiated cell है, specialized cell है, उन्हें आगे फिर से division की जरूरत पड़ जाती है. Like in this case, you can take an example of the secondary growth in the diploid stem. जो diploid stem होते हैं, diploid stem के अंदर secondary growth होती है. At that time what happens, एक रफ से डायग्राम बना के यहाँ पे समझाने की कोशिश कर रहे हूँ कि क्या होता है इसके अंदर. This is the epidermis of the stem and here the cortex is present and this region, cortical region is formed from the parenchymatous cells. ये parenchymatous cell से बना हुआ region है. Parenchymatous cells means these are the specialized cell. ये specialized cell है. मीन्स मेरिस्टेमेटिक नहीं रही. मेरिस्टेमेटिक सेल्स जो है पैरेन्काइमेटस सेल बन गई है, पैरेन्काइमा टिश्यूज में कन्वर्ट हो गई हैं एंड दे बिकम द स्पेशलाइज्ड सेल. इस स्पेशलाइज्ड सेल में कन्वर्ट हो चुकी हैं. नाउ व्हाट हैपेंस, दीस पैरेन्काइमेटस सेल्स, इनमें से कुछ पैरेन्काइमेटस सेल हम ले सकते हैं. सपोज these parenchymatous cells now become meristematic again. ये जो parenchymatous cells हैं, they will become meristematic so that they can form the cork cambium. So during the secondary growth in the diploid stems, to make the cork cambium, some parenchymatous cells become meristematic again and means they have to come back into the cell cycle. उसके लिए क्या होगा, Z0 में सेल एग्जिट कर गई थी सेल साइकिल से, बट इफ इट नीड टू बी रिक्वायर, इट नीड टू डिवाइड अगेन, फिर से डिवीजन की जरूरत सेल को पड़ रही है, फिर से सेल को मैरिस्टेमेटिक सेल बनना है, डिवीजन स्टार्ट करना है, देन इट कम बैक, इट नीड टू बी कम बैक इनटू द सेल साइकिल अगेन. तो इसी सेल साइकिल के अंदर फिर से एंटर करेंगी एंड दीज सेल्स which come back into the cell cycle to become meristematic, or by becoming meristematic, this condition is known as de-differentiation. The second condition is known as de-differentiation. So this is the another condition. The first condition was the differentiation. डिफरेंशिएशन हम लिख सकते हैं, डिफरेंशिएटेड तो सेल हुए, डिफरेंशिएशन, the cell do the differentiation. When they become specialized, then we call it differentiation. When this differentiated cell or specialized cell which exit from the cell cycle and they come back again into the cell cycle, they come back into the Z1 and then they do S and Z2 and after that they need to be divided. अगर फिर से सेल साइकिल में एंटर कर जाती है, again they become meristematic cell, then we call them de-differentiation. These cells are known as de-differentiated cell and this process is known as de-differentiation. Okay, this is the one condition. Okay, यहाँ पे हमने देखा for the de-differentiation the meristematic cells will become the parenchymatous cell will become meristematic cell again. यहाँ पे parenchymatous cell जो थी वो meristematic cell बन गई हैं for the division so that they make the cork cambium. Now let's see what happens. One more condition is there. एक और condition create होती है जिसमें क्या होता है कि in the cork cambium हमने देखा कि जो cork cambium बन रहा है, this cork cambium जो बनता है secondary growth के अंदर, this cork cambium will make the cork, ये cork बनाएगा towards the outer side की तरफ cork form करेगा and towards the inner side it will form the cortex, secondary cortex form करेगा towards the inner side. During secondary growth the cork cambium will form the cork towards the outer side and secondary cortex toward the inner side. For this it needs to form some parenchymatous cell and इसके लिए यहाँ पे parenchymatous cell जो होंगी produce होंगी. Means we can say that in this condition the meristematic cells which further they were parenchymatous cells, they convert into the meristematic cells and after that they will again form the parenchymatous cells. And this condition where the means this de-differentiated cells again become the specialized, they will again exit from the cell cycle. This condition is known as redifferentiation. So what happening here that cell, this cells which is entered here, ये enter किया था and again it is exiting from from the cell cycle, फिर से cell cycle से exit कर देगी. Then we call it redifferentiation. This process is known as redifferentiation, and the cell will call as redifferentiated cell. Let's see what happened here in the normal cell cycle. Cell passes through the interphase and then it enters into the M phase, where it divides and form two daughter cells. These daughter cells will again enter into this interphase and continue the cell cycle. But when a daughter cell is formed and it do not want to divide further, it want to become a specialized or differentiated, then they exit from the cell cycle. तो वो cell cycle से बाहर निकल जाती है, exit कर जाती है, and this condition is known as Z0. These cells are known as differentiated cells, and the phase of the cell is known as Z0 phase. इस phase को हम बोलते हैं Z0 और Z0 phase. This is the inactive phase for the cell division, but all other activities will remain continue. इस Z0 phase में cell rest करेगी, inactive रहेगी, कोई division नहीं होगा. But at some times this specialized or differentiated cells will enter into the cell cycle again. फिर से cell cycle में enter करेगी so that they can divide again. तो फिर से division कर सके, हमारे somatic बन सके. At that time we call these cells de-differentiated cells. उस time पे इस cells को हम बोलेंगे de-differentiated cells, and the process is known as de-differentiation. And later on, if these de-differentiated cells need to exit from the cell cycle again, they need to become permanent or specialized again, then we call these cells redifferentiated cells, and the process is known as redifferentiation. So here we discussed some exception again other than the normal cell cycle. ये एक तरीके से कुछ अलग phases हैं जो normal cell cycle से अलग होती है. इनके बारे में हमने यहाँ पे बात की है. After that we need to discuss about the DNA content, or we can say the DNA content or chromosome number, which is also known as C value of the cell. So in the next video we will discuss about C value.